Now this whole area is one of extremely high interest, as we can see on this first slide. Basically, as you may know, in January of this year, President Obama announced that we're going to have a new precision health initiative, and the exact nature of this initiative is not known, but it will involve being able to bring genomics and other omics activity into better being able to manage people's health and ultimately help treat any diseases they may have. What frames our thinking a lot in this area is shown here. Basically, your health, it's due to a combination of things. So we like to think of health as a state where you'll toggle between healthy periods and disease periods, as shown in the center of the slide. And then many factors will contribute to this. And I think we know this intuitively. You have things like your DNA, shown in the upper left corner, and then other parameters as well. So for example, the various things you're exposed to, the so-called exposome, that would mean the food you eat, pathogens you might be exposed to, any life stresses you might encounter. It also means your exercise and activity and things of that nature. So all these together, as I say, they do contribute to your health. And I think we know this in intuitive terms, but I would argue one goal of many of us is trying to understand this in quantitative terms, so that someday when we sequence people before they're born, you'll have a genome sequence, and you'd like to understand that in that context, with other sorts of exposures, what kinds of health outcomes you might expect. And so this is really the goal, to try and understand this in probabilistic terms, if you will, and get very quantitative about our predictions. So the one part of this is easily solvable now. This is your genome sequence, and that's because of the revolution in genome sequencing that's occurred in the last decade or so. And that's shown here. In the last, as I say, decade, basically the cost of genome sequencing, it's been dropping roughly tenfold every 15 months or so. And it's now at the point where we pay about $1,400 for a research genome. So in fact, that is getting affordable for many people. However, even though it's $1,400 to do the sequencing, it's still very, very expensive to do a proper interpretation, at least for a healthy person. And indeed, it costs us about $15,000 to interpret a genome the way we do it, because we look up a lot of variants manually to make sure everything's correct. I think that cost will drop a lot too, but there's still ways to go. Nonetheless, it is possible to get your genome sequence, and genome sequencing is now becoming very popular in the clinic. And if we can add the next slide, basically this is just some of the areas in which genome sequencing is having impact on medicine. So basically, as you'll see at the top, genome sequencing is having high impact in the area of cancer. If you have cancer, in many cases it makes a lot of sense to get your genome sequenced so you can see what kinds of genetic changes are there, and some of those actually may be actionable in terms of drugs being able to target the changes that have occurred from those genetic changes. Other areas where genome sequencing is having high impact is in solving mystery diseases. There are many undiagnosed diseases that exist. About actually 0.4% of kids are born each year with a serious disease that's not solvable, and people are turning to genome sequence to try and actually understand the underlying cause. And it works in about a quarter to a third of cases now. People are able to solve what's wrong with their child through genome sequencing. It's often not actionable, but it's still very useful for parents to try and understand what's wrong with their child. It's actually very useful information to have. And lastly, genome sequencing is having huge impact on prenatal diagnostics, because as you may know, there are actually tests where fetal DNA does circulate in a pregnant mother, and you can actually extract the DNA from the blood of a mother and sequence the genome. And there are FDA-approved tests to look for aneuploidies in this area. Now, the area that interests us the most is at the bottom of the slide, and that's where we're going to spend a lot of time. And it's this idea that if you're a healthy person, can genome sequencing and other sorts of omics technologies be useful? And that was certainly an open question when we entered this area about 6 years ago, and decided to see if we could actually bring genomics and other sorts of omics to the clinic. And so around that time, we set up something we call personal omics profiling, which is shown here. The idea is that in addition to your genome, you would get many other sorts of measurements as well. You would in fact get measurements about your epigenome, and specifically that means DNA methylation. We'll talk a little bit more what that means in a minute, but the idea that non-genomic things can actually influence your health. You might be able to pick up with epigenome measurements as well as measurements on all your RNA, that's your transcriptome, all your proteins, that's your proteome. The way we do it, we miss certain kinds of proteins. We use mass spectrometry typically to follow the proteins, and that misses some of the more rare proteins that are very important. Cytokines are certainly important, so we follow those as well. In addition, we follow antibodies you make against yourself called autoantibodies, and we have chips that let us follow all our autoantibodies as well as all viruses that are known to infect humans. Those are down on the array as well. And then lastly, we have the microbiome. We follow 5 different microbiomes, and we'll talk about that a little bit more in a minute. We follow the microbes that live in your gut, in your nose, your nasal, urine, skin, and tongue, 5 different microbiomes. And so basically, initially, we didn't have the microbiome or the DNA methylome, and we were following about 40,000 or so molecules. The assays were a little more primitive. These days, we literally make billions of measurements every time we sample someone. Now, just to get this project started, we picked a person who was readily accessible and easy to work with, and of course, that is me. So we actually started profiling me, and I'll show you the outline in just a minute. But I forgot, I wanted to actually mention why are we doing all this? Why do we want to dig in so deeply? And there's a number of goals to the study. First and foremost, we want to see how individuals change over time and as they go through healthy and disease states. We actually follow people and sample their blood over time, and you'll see in a minute we've been following me for quite some time. We also want to understand how the different omes relate to one another. It's the DNA methylome and the transcriptome and the proteome and the metabolome, and which of these is in fact the most useful, and what kinds of information can you get from each of these omes and how do they relate to one another. Other sorts of things we want to follow are how individual responses are similar between people and how they differ. If we really want to execute personalized medicine, we need to understand individual differences and why some people behave a certain way when they're faced with viral infections or other perturbations, and why other people are essentially unaffected. That's going to be very important. And then lastly, these detailed profiles we do because we hope to learn information that will basically be able to help manage people's health better. So we may learn useful information that will help us predict the risk for certain diseases and help them manage disease. So as I say, the way we launched this project was we just chose me to get the technology working, and we've actually been profiling me for over 5.5 years right now. So we started this in 2010, and we've taken over 120 different time points, sampled over 120 times, actually for the microbiome even more than that now. And during this period I've been through 8 different viral infections, all common colds. 6 of them are shown here. So you'll see in red are the rhinoviruses, in green is respiratory syncytial virus. That's a common cold in children, not so common in adults, but they do get it. Usually they get sick when they get one of those. Adenoviruses, I have had 2 of those so far. And then most recently, in fact, I've even had Lyme disease. So I've gotten yet another disease. So we've been able to profile me through those times. So basically what we do is we take blood and analyze all those omes, if you will, at healthy times, so every 2 to 3 months when I'm healthy. And then whenever I get one of these viral infections or at interesting times, we do much more dense sampling. So you'll notice under the RSV infection, there's about 7 different time points we've sampled during that time, again, to see the dynamic changes that occur when someone gets sick. So just to summarize some of the first part of the study, we started by actually sequencing my genome, which made many predictions about my health, most of which I knew from my family history. So that is to say, this is just showing one part of the analysis, my analysis of complex disease. But to illustrate the point, that the stars are some of the areas about problems I knew I already had or was at risk for. So as an example, everyone in my father's side has had some sort of heart problem, so being at high risk for coronary artery disease made a lot of sense. It's also known that I have high cholesterol. I knew that already, and so that shows up as that hypertriglycemia and the lipidemia. So you can see I'm at increased risk for that. Those are the orange lines to the right. I'm at low risk for obesity. No one in my family's ever been overweight. That's the blue line to the left, so it shows I'm at much lower risk. So these are the sorts of things my genome predicted that I was pretty much knew already from family history and other measurements that have been taken. But there were several new things to be learned. One is that I was a very high risk for basal cell carcinoma. And it turns out that actually was in my family, I just didn't realize it. And the way I discovered that is they were all out visiting, and we basically took blood from all of them so we could profile them, of course. And then I also talked to several, and in fact, some of my family members have had basal cell carcinomas. It's just that it wasn't known to everyone in the family. It just wasn't discussed as much. So, in fact, those lesions were there. It wasn't my family history, but I just didn't know it. And what I like to say is that, in fact, your genome is a better record of your family history than your family is, because if you know how to interpret it, the information is there. The other thing that came up early on in the study is that I was a very high risk for type 2 diabetes, going from something like a 26% chance, a 47% chance of having type 2 diabetes, and that was a surprise to me. I was not aware of that from my family history. But we had been, in fact, watching my glucose and profiling during time, and as shown on the bottom of the slide, in fact, my glucose had been going along quite normally in a very normal range. But ironically, when I went in to get a very fancy glucose test, you'll see that's where the bar starts going up. That time point in the middle is the day I went in for that test. And that's just my fasting glucose level. It came out very high, 127. That's not normal. That's higher than it should be. And we were both surprised. We repeated the test, came out high again. Measured things a week later, and you can see it's even higher if you look at the next time point, 147. We also measured my hemoglobin A1C at that point, 6.4, which is right on the edge of being classified as type 2 diabetic. So turns out for diabetes, if you have a measurement of 6.5, you're classified as diabetic. And I was right on the edge. So 5 weeks later, I measured myself, or got measured again, this time through a general practitioner, and my glucose came out even higher, hemoglobin A1C, 6.7, classified as diabetic. So I can tell you when that was April 11th, 2011, because up until then I had a very bad diet. I used to eat lots of desserts and sweets because I didn't know I had a problem. So I cut all that out, and I had been biking, but I doubled my biking and actually basically cut out all the excuses for not biking. And then also started running, and it didn't happen right away, but gradually I could bring my glucose levels down to normal. Now, all this made a big splash at the time because it's the first time someone ever used their genome to predict they're at risk for a disease and got the disease. And in my case, thought it was very manageable. It's also of high interest because you can see when all this happened, it happened right around the time of this nasty respiratory syncytial virus infection, the RSV infection. That's when things went off stray. And people have shown viral infections are associated with type 1 diabetes, not necessarily with type 2. So this was a new finding. And so the hypothesis is that my genome has me predisposed for type 2 diabetes. And then in conjunction with the viral infection, that's what triggered the disease. So a combination of genetics plus environment causing this disease. Now, some people criticize and say, well, maybe that disease would have gone away on its own. And so we accidentally did a control experiment, if you will, the right experiment, which is, it turns out I had stopped running. After I got my glucose down under control. So on the left is that glucose spike and the hemoglobin A1C elevation. And then I got both of those under control. But ironically, after I stopped running, and there were some other things that happened too. I got a rhinovirus infection and I switched statins. Any one of those might be contributing to this, but the bottom line is my glucose went up again pretty rapidly, it looks like, and was actually quite high. And what was interesting is that we were collecting all this data, no one's looking at it because you'll see in a minute we scaled the study to study 100 people. And basically what we discovered is that the glucose was high, it was very high. You'll see that right spike where it actually in fact got to 7.0, and that's when someone looked at the data and said, oh my goodness, Mike, your hemoglobin A1C is 7.0. Did you know that? And I said, no, I had no idea. So I've been running high for about a year, and then basically I sampled myself a few times, and then started exercising. Yeah, I can reduce it some, but it's the best I've been able to do is get it down to 5.7. And then lately, I had Lyme disease this summer. It's been creeping up. It's right around 6.4 when I last looked. So I'm back on the edge. So the bottom line is I do have a glucose metabolism problem. You can see it's very elevated. And it's not just me. It turns out my brothers and sisters are all elevated for their hemoglobin A1C too. And they're just as thin, if not thinner, than I am. And so, again, there's no question we have a metabolic disorder running in our family. And this was all discovered initially through the genome sequence. And then making the other measurements was very useful for finding this as well. So this is where we are right now. In fact, I may wind up taking metformin to try and bring my glucose levels better under control. Now, in addition to measuring glucose, remember we were measuring lots and lots of other things, including all my proteins, my RNA, my metabolites. Again, initially making 40,000 measurements, now literally billions of measurements. And we can follow in a very systematic fashion the molecules that go up and go down at interesting times. So say times during viral infection, or in this case, what's shown on this slide, times when I've had, I got the type 2 diabetes. And what you'll notice if you look at the top panel, in green are all the molecules. This is, they've all been combined in this analysis where we've combined transcripts and proteins and metabolites. And in green are all the molecules that go down. In red at the top are all the molecules that go up. And then we also look for spikes of things that happen. And what's shown here is, in fact, again, across the RSV infection when I got type 2 diabetes, on the left I was at normal glucose levels. The third lane is actually when the RSV infection hit. And then, as I say, things started going off around that time for the sugar levels. And at the right side, the levels have decreased, the glucose levels have decreased some, but they're still somewhat elevated. I am healthy, but I have somewhat elevated glucose levels. And the net result from the top panel, when you look at all the things that systematically went up or down, you can see what kinds of pathways change. And in fact, my insulin biosynthetic pathway has dropped. It's in the green section there, and at least the molecules are. And basically, my insulin levels on average have never been as high as they were before the RSV infection. So we think that's one of the things that's come out of this. The other is we do see spikes of things happening. So at 2 weeks after the RSV infection, a little bit over that, we see interesting pathways changing. There's a burst of stuff happening. So one of the top things changing is glucose regulation of insulin secretion. And one of the pathways that goes down, that's in the bottom pathway, is platelet plug formation, etc. So we can see in incredible detail the kinds of changes that occur when people get disease with this very detailed omic analysis. So what I like to say is that I think this is like getting an IMAX movie, if you will, of what goes on when people get disease. And of course, if the way it's currently measured, if you think about the way we practice medicine now, you measure about 15 different things. And I would argue that's like fuzzy radio at best. So this is a much higher resolution picture of what happens as people get diseases. Now, since this first study, we've added on many other sorts of things that we have not yet published, but we think are going to be very powerful. One of those things is following my DNA methylation pattern, which is basically an epigenetic change, if you will. You may have heard that term before. But basically, your DNA modifications, this is a chemical modification that occurs on DNA, and it's affected by nutrition, lifestyle factors. So, for example, exercise is known to affect methylation patterns. Your age, you can actually tell someone's age from their methylation pattern, and I can tell you from mine, it works quite well. It's very precise. And other environmental factors are thought to affect your DNA methylation patterns. And DNA methylation has, at least in the promoters and in the gene itself, have been associated with gene silencing. So we've been able to map my methylation sites across the genome by a technique called whole genome bisulfite sequencing. This has been done for 40 different time points. And basically, we find all kinds of interesting stuff. I'll tell you one interesting vignette, if you will. We've gone through my genome and looked for cases where I might have genes that would be differentially affected, one by genetic mutation, one by DNA methylation, and whether any of these genes might be associated with human disease. And it turns out I have a number of such cases. This is one example shown here. This is the PDE4 gene, which is involved in eosinophilia, as you've seen. If you have mutations there, it can affect your eosinophil numbers. So it turns out this is a case where one allele, in this case my father's allele, has been inactivated by mutation, or so it's predicted from the genome sequence. But it looks pretty clear that that is an inactivating mutation. And my mother's allele is actually methylated and chemically modified, and that gene, we believe, is turned off. And consistent with this, we've done RNA-seq measurements, and I have very few reads, if you will, from my mother's. Out of 47 reads, only 3 come from my mother's copy, and the remainder come from my father's copy, meaning my father's gene is still expressed, but it is mutated. So this gene, if you will, it's been inactivated by 2 different methods, one by genetic mutation, one by modification, and it, in principle, could be contributing to human disease, so it's something I want to keep an eye on based on this information. So what I would like to say is that it's not just your genome that's important, but it's also your epigenome, and I think we need to ultimately be able to interpret both to be able to better predict human health and disease risk. The other area that we've been looking at a lot is, in fact, the human microbiome. So you may know that you have many more cells in and on you that are not you than are you. That is to say, we have 30 trillion cells that are human cells that are us, and we have 10 times that amount that are actually bacteria and other things that live in our gut and on our skin and all over us. And they're very, very important microbes because they actually digest your food. The gut bacteria digest your food. It makes essential vitamins that you absolutely need to be healthy and survive. And so it's very, very important for your health. And these days, the microbiome has been implicated in every human disease that you can think of. Crohn's disease, diabetes, autism, what have you. Although in most cases, what's cause and effect isn't so clear. There is an association, but it's not clear the microbiome is causing it or responding to the disease state. Although in some cases, like diabetes and obesity, it probably is. There are direct links that have been described. So there is more likely a causal relationship, at least in some cases. So basically, about 3 years ago, a little over that, we've added on the microbiome, initially on me, now you'll see on this bigger cohort, and we've been analyzing folks. And when we first started this, we decided to analyze one of my interesting time points that's shown here. This is, if you look at the circle, this is a viral infection. What started out as a rhinovirus infection, and then something like 9 days later, a secondary infection occurred. And we took all kinds of swabs and could not figure out what was wrong with me. Okay, so that basically got, if you will, put on hold. It was just left as a mystery infection. Well, when we started doing microbiome analysis, this is the first time series we looked at, and we looked at both my nasal and gut and other sorts of microbiomes. And as shown here, basically this is looking at my nasal microbiome. Basically the left column is when I'm healthy. The second one is when I'm sick. The third column in the middle is 2 days after getting sick. So I should say that's sick with the mystery infection. Then 2 days later, then 2.5 days later, and back to healthy. And there's one bacterial strain circled there, Streptococcus pneumonia, that correlates perfectly with when I got sick. So the odds are I got a strep infection, and this is probably the most expensive way to have figured this out. So that is to say, it's a very expensive way to figure out you have a strep infection. What's kind of interesting about all this, though, is we not only looked at my nasal, but we also looked at my gut microbiome, and that's shown here. Basically, same time points as before. It's displayed a little bit differently, but on the left are my healthy samples. Again, you see the fever one, and then as I'm recovering. And in general, the healthy states on the left and the ones on the right look pretty similar, not identical, but pretty similar. And then you'll see that my stool microbiome, I forgot to say, this is my stool or gut microbiome, it actually shifts pretty dramatically. So what we think is when you get sick, it's not just one isolated area of the body, but basically you undergo a systems-wide change. And by making all these measurements, we can see this in detail. And I'm not saying they're direct. That is to say, it may be the food you eat that's different. Could be the fact that you have mucus flowing into your gut and such that may shift your microbiome. But the point is your whole body does change in response to various disease states. And so I think we need to think about that as we're treating individuals. So we haven't just stopped there. We're also adding other kinds of information as well. So we've been adding on a lot of other physiological and activity measurements. So you may know that there are smart watches out there. I wear multiple watches myself that measure different things like sleep and heart rate and steps. There are also free applications on your iPhone that do this too. And so I have a number of applications on my iPhone that does this. You can even measure your electrocardiogram on your iPhone. We do things like continuous glucose measures on folks, including me, so we can follow what their glucose levels look like with the various foods they eat and so on. So we can collect a lot of other information and be able to see in detail the kinds of things that people are doing with the ultimate goal of relating this to the biochemical changes that we observe from the omics profiling. So we also haven't, as I've alluded to, we haven't just focused on me. We've been able to enlarge the study, and just to give you a few vignettes here. So basically, we've now carried a number of sequencing studies of healthy folks. Up the top is a case where, just a few examples here. The top shows a case where we sequence one family, and both the father and daughter are shown to have a factor V mutation, which affects blood clotting. And actually, this kind of information we would argue is very useful, and very useful to her for helping her decide what kinds of birth control she might want to use. We also sequenced with Ewan Ashley and Tom Kutermis a set of healthy folks, initially 12 we did, that we sequenced, and we did see disease risk mutations, but one very important one we saw was a woman who had a BRCA1 mutation. No question, it was an inactivating mutation that disrupted and actually clearly killed the gene. And based on this information, the woman actually opted for surgery. And so we would argue having this information is super important. And what you decide to do with that information is your decision. You may not want to have surgery, but you certainly would want to get yourself monitored. And I forgot to say, but she did not have any family history of breast cancer. This is very, very valuable information to have. She would not have discovered it had we not sequenced her genome. Now, in addition to genome sequencing, we're also doing these personal omics profiles that we've been doing on me. And we're now doing it on another 100 folks. We started the study a little over 2 years ago, some even before that. And we've been profiling them just like me, where we sample them every 3 months or so when they're healthy, and then do very dense profiles when they get sick or undergo some sort of stress response. So for example, some individuals were up for 48 hours without sleep, and so we sampled their blood right after that and things like that. So we can see interesting changes, and you'll see in a minute, we've also looked at folks who have been over eating and changing their diet, and I'll tell you a little bit about that in a minute. So far, this is a bit of a progress report, but we've collected over 850 time points from this cohort. Obviously, the most from me, from the top line up there, but we have over 50 people where we have 5 or more time points. We're getting nice longitudinal information from this group. And one nice thing about this particular cohort is that they've all agreed, with a few exceptions, to make their data completely public so that the scientific community will be able to access the information and be able to better study the data and try and understand what's going on as people go through healthy and disease states and various stress responses. To give you a few vignettes of what we've learned, we're learning a heck of a lot already. But this is one example of something we've learned just looking at the genome sequences. Just like I mentioned before, we've already found some individuals. We've sequenced pretty much all 100 or close to all 100. And we've gone over about 15 people's genomes just like mine, where we go over very, very carefully. We've found some interesting mutations, just a few examples that are shown here. One person had a mutation in that SHDB, which puts them at very, very high risk for paraganglioma. Another person has a protein C mutation, which, like the factor V mutation, affects coagulation. These are very valuable pieces of information for them to know. And of course, everyone has carrier mutations for certain diseases, and we all have very interesting variants that affect your drug responses. So for example, I have variants that affect my response to warfarin, which you would take in the event of, you might take in the event of a heart attack, and my genome tells me what dose to start with. It also tells me what statins I should avoid. I am on statins for my cholesterol, high cholesterol. My genome tells me which statins I should avoid. To give you a few other stories, we've been looking, a set of our folks have been undergoing what's called an overfeeding study, where they basically eat an extra 1,000 calories a day, at least for the men, 750 for the women. So they eat these extra calories every day for 30 days, and they gain weight, 6 pounds on average, and then they diet for 60 days and they go back to baseline. So we sampled them at the beginning, at peak weight, and back at baseline. They're part of our study, so we're following them throughout. And then again, we measure what kinds of changes are going on. So far, we've analyzed about 24 folks. 13 are insulin resistant, which is a precursor for type 2 diabetes. I forgot to say that our cohort of 100 people, about 2/3 are pre-diabetic or insulin resistant. They have some glucose control problems. The rest are healthy, generally BMI matched. So for this particular sub-cohort, 13 are insulin resistant and 11 are healthy controls. Some of the fun things we found just right from the start, we've discovered that even at baseline, before these folks have gained weight, the insulin resistant and the insulin sensitive folks are in fact quite different. This is the RNA-seq data, but we could show this for other examples you'll see in a minute. But the insulin resistant group, they're elevated in their expression for a set of Mendelian genes called MODY genes that is involved in beta cell production and glucose control. It's a somewhat diverse group, not always well studied for some of the genes, but anyway, it has been associated with more what's called Mendelian forms of diabetes, oxidative phosphorylation, and interestingly, the platelet genes are elevated in insulin resistant, and other sets of genes are higher in the insulin sensitive. Now, when they undergo the overfeeding, we do see dramatic changes. In general, everybody goes up for the inflammation response on average, down for the platelet-specific genes. Very fascinating. Those genes actually are showing interesting dynamic changes during metabolic times, like overeating, or in my case, when I got diabetes and things, they keep popping up. Anyway, when you lose weight, that does reverse. The inflammation response goes down. So what this is saying, that if you eat a lot, you actually undergo a pretty serious immunological, or at least a somewhat strong immunological effect. And so that's clearly one of the major things happening for folks who overeat a lot fast. I won't get into this, but we can see similar kinds of changes for metabolites as well. People are very different. Our insulin sensitive, insulin resistant folks are different even at baseline. And again, they will undergo dramatic changes as they overeat and lose weight. The other sort of thing we've discovered is that even at baseline, when people are healthy, they can be very different in the kinds of molecules they have expressed. And this shows one example here. This is looking at 63 cytokines in 20 people. Each color is a different person. And what you'll notice, I've flagged 2 examples here with red arrows, but what you'll notice is that some individuals are higher levels of some cytokines than the group on the whole. And so everybody's a little bit different in their profile. So we can see this for cytokines, we can see it for RNA, we can see it for lots of different things. Again, this is at a healthy time point. So we're all poised differently with the kinds of molecules that we have expressed, and potentially this does affect our susceptibility to disease and such. And so one other example of this is in fact the microbiome. So this is known already, but everybody has a very unique microbiome. What we've discovered is that when people overeat or go through viral infections, they will shift their microbiome, but nonetheless, at the end of the day, they're still closer to themselves than other people who have a very similar perturbation. So that is to say, if a bunch of people get sick, they will shift their microbiome, but they still look closer, again, to themselves in terms of microbiome rather than to the other folks. Now, there are some commonalities, but those are just as a general what we're seeing. I can tell you this is still early days. There is some correlation between what we see in the metabolome and the microbiome. That's something we would expect. It's nice to see. And this slide actually illustrates some of that shown here. The ultimate goal of all this is really shown on this slide. We're really trying to understand at an individual level how some people, when exposed with certain environments like food, or some people at the top, you'll notice they gain weight, they become insulin resistant. Other people remain perfectly healthy, and we want to understand why that is, why some people do one thing and others have bad things happen. We also want to understand, for example, in the case of viral infections shown at the bottom, why some people get sick and others stay healthy. And as an example of that, plenty of people get RSV infections, don't get type 2 diabetes, but it sure seems that was the case for me, since that strong correlation. Ultimately, where I see this going is illustrated here. I envision a world where people will get their genome sequenced before birth in the circulating DNA in the mom. And then we will combine that with other sorts of information. A lot of it will be the activity information that will all relay into your iPhone, but there will also be other biochemical measurements that get made at a much higher frequency, and many more things will probably get measured in the future. And we'll use this information to be able to better predict risks, diagnose disease early, and ultimately treat and manage disease. So in summary, what I've told you is that personal genome sequencing is here. It can be used to predict disease and manage health. We would argue these multi-omics profiles are extremely powerful. They give lots of information. They help see much, much better about what's going on. And ultimately, we hope they'll be able to better help us teach us how to treat disease. I think the other thing that's very, very important about this study is that this is a longitudinal profile. It's important to see how people change. And if you think about it, at the end of the day, you really want to see how your altered state compares to your healthy state, and not everybody else's healthy state. So I would argue you should get your healthy profiles done now while you are healthy, so that if something bad happens, you will see the altered state. You'll be able to better interpret it relative to your own healthy baseline. Another important point is that we are all different. We all have different biochemical measurements like the cytokine and the microbiome, and this is very, very important. So lastly, I do think people will ultimately be responsible for managing their own health better because it is complicated, and this information can feed back directly to you at some level, that is from the wearables and potentially some of the other tests. And I think people are going to have to take charge of their own health, because physicians of the future, they'll be able to guide you and coordinate your health care, but I don't think they'll be quite as well positioned to manage your health as you are. So overall, this project was started by Rui Chen, George Mias, and others. And as we scaled up, this is the group shown here. Wenyu Xiao, Brian Pining, Kevin Contrepois, Tejas Mishra, and many others have been instrumental in my lab for setting this up. And we also have a wonderful collaboration with George Weinstock's lab, who's been doing the microbiome, and Tracy McLaughlin has been doing a lot of the clinical and patient recruitment part. And so it's really been a wonderful team effort to be able to carry out this project. For those more interested in this kind of general area, I'll just point out that we actually have launched a genetics and genomics certificate program that talks about cancer genome sequencing, personalized medicine, a lot of the things here. This is an online class that you are welcome to take if you want. You can visit the website shown there, geneticscertificate.stanford.edu, and you can learn more about some of the topics I just described, as well as many other topics like the ones I said, solving undiagnosed disease, cancer, and what have you. So thank you very much for having me here today. The biggest challenges are clearly who pays. It's great to roll out these technologies, but we need someone to pay for them. And that's particularly difficult when you're thinking about trying to keep people healthy. Our system's very well set up towards treating people who are sick and have diseases like cancer or even some of the undiagnosed disease, but it's not as well set up for paying people to stay healthy, and this is expensive to roll out. I mentioned it before that it's about $1,400 to sequence a genome, $15,000 to interpret it. when it's a healthy person's genome, and someone has to pay that, and if people are always changing their provider, as occurs in the US, there's some reluctance on insurance companies to pay for that if you're going to be switching to another company in a few years. So that is a challenge that may go down as health changes, as costs drop. Now the other thing I can say is I actually think insurance companies should be paying us to get our genome sequenced, because I actually think you will be managing your health better with this kind of information. Much like me, based on my genome, I was actually able to stay on the alert to be able to look out for diseases, and I think that'll be true in general. Having this information will be very, very valuable. So that's probably one of the biggest challenges. There are other challenges as well, like interpreting genomes. We still are at an early stage. We have to get better at it, but I think we will over time. And those are probably some of the biggest challenges. Well, I think systems biology will mostly be helpful for just managing your health. That is to say, with all this information, we can better interpret people's health states. I believe it's hard now, but I think we will get better at it in the future. Certainly genomics is helping. And I think there are strong cases about genomics plus transcriptomics helping manage people's disease in the case of cancer. So it's a no-brainer we'll be adding at least some of these other sorts of omics in the future. And they will better, and to give you one example, would be cancer. That is to say, if you look at driver mutations in cancer, every time cancer occurs, there's typically multiple mutations. But when you sequence people's DNA, there can be up to tens of thousands of mutations in the genome, and trying to understand which ones are truly drivers is hard. But the transcript actually rules out a lot, because a lot of those genes aren't expressed. It turns out 2/3 of them aren't expressed. So by, in fact, doing the transcriptome, you can see which mutations are really most likely to be driving cells to grow uncontrollably, and that'll be valuable. So these are the ways in which it'll be useful. I think it'll also then, with that kind of information, you'll also make sure you're applying the right drug to the right disease. So the better you can diagnose the disease, whether it's cancer, whether it's undiagnosed disease, the more you can give them the right therapy. And again, for cancer is another great example. This is really probably the most mature in this area, but some drug treatments cost $100,000 or more for cancer patients. And it would be terrible, of course, to be treating people who have no chance of responding to those drugs if you're just treating them if they're not going to respond. So we really need to figure out who responds and who doesn't, and genomics and systems biology will be a better way of figuring this out. Yeah, there's no question that big data is very challenging. Just on me alone, I have half a petabyte of data. So managing and analyzing this is not easy, especially right now, because a lot of this is research, and we don't know the best way to analyze the data and bring it out in a useful fashion, either for research or for clinical use. Although I think we ultimately will get better. That is to say, you will see people collecting lots of data, and when it gets analyzed, we will be able to find common themes out for research. Now, I do think it's important that we share the data. It does get tricky because there are privacy concerns and things like that. But I do think it's important we get these data into the public domain for research, so that people can actually access this and share information. And that is the power of big data, that if we have large pockets of data all around the planet, and some people are responding to drugs and some people aren't, we can use that data to better understand which drugs go to the right person. So I do think there are challenges with how to do this in terms of managing privacy, but I think most people are very eager to see it happen. Most people would rather solve diseases than worry about their privacy. So I actually think the privacy thing is a little bit overplayed as well. And then I think when it comes to bringing big data to the clinic, there is that aspect I said, by actually having a lot of data out there and seeing who responds and who doesn't. You can use evidence-based information, and real time, actually. You should be able to have algorithms that say, as of today, this many people with that kind of a genome sequence responded to this drug and this many did not, and that will be very, very valuable. I do think for clinicians this can get tricky. For your regular physicians, they're not going to know, that they don't have the analytics, nor necessarily should they, to be able to calculate things and bring it. So I think this has to distill into a very useful, simple report for people, and it's not unlike what happens for imaging now, where basically specialists really interpret the images and write up a report which goes back to the physicians. That's how I see big data of the future rolling out too, even though there'll be lots of analytics, lots of things, people poring over lots of data. At the end, it'll turn into a simple report, like here's what your genome sequence predicts about your risk, here's what we learned from this omics profile that can be used to help treat a person. So I do think, again, the key messages are sharing data worldwide so that we can do the best evidence-based medicine we can, and then distilling it into a simple form so that physicians can best relay this information back to their patients. I'm going to